In this mini lecture, we're going to take a look at the organic molecules that are found within cells, and specifically we're going to concentrate on the carbohydrates. The next mini lecture associated with this series is going to take a look at the organic molecules known as the lipids, the proteins, and the nucleic acids. The chemicals that are used in metabolic reactions, or those that are produced by metabolic reactions, can be classified into two groups. We can classify them as inorganic molecules and organic molecules. In the previous lecture, we looked a little bit at inorganic molecules. Specifically, we took some time to take a look at water. But in these lectures, we are now going to explore the organic molecules. Believe it or not, defining what an inorganic compound is kind of difficult. But I'm going to give you the easiest definition. An inorganic molecule is a molecule that is not organic. And these are things like water, oxygen, carbon dioxide, inorganic salts. And I'll tell you why they're inorganic, because they lack carbon-hydrogen bonds. If you are a compound or molecule that contains a carbon-hydrogen bond, you are an organic molecule. However, you also have to be derived from a biological source. So the actual definition of an organic molecule is a molecule that contains carbon-hydrogen bonds and is derived from a biological source. You can have additional elements like oxygen, nitrogen, phosphorus, and sulfur. You can attach those to that carbon-hydrogen backbone, but you've got to have the carbon-hydrogen backbone. At the basis of every organic molecule is carbon. And carbon has several properties actually that makes it pretty useful to living organisms such as human beings. Each carbon is going to bind to 4 other atoms through covalent bonds. This means that carbon can form bonds with other carbons to produce very large molecules that can have many different unique shapes. Atoms that bond with carbon through single bonds tend to produce a molecule with a pyramidal or tetrahedral shape. Long chains of carbon-based molecules become possible when you string one carbon to a neighboring carbon to a neighboring carbon to a neighboring carbon. In molecules with multiple carbons, the type of carbon-to-carbon bonding is really important in determining the molecule's shape. This is because single carbon-carbon bonds tend to produce a linear bond. And what you end up with is kind of a linear chain of carbon with the other atoms that sort of project out. So if you take a look at this figure here, right in the center, ethane, you can see the 2 black circles. Those are the 2 carbons. Notice they are in line with one another. And then the hydrogens sort of project out in a 3-dimensional manner. But when you take that carbon-carbon single bond and turn it to a carbon-carbon double bond, look what happens to the other atoms bound to that carbon. You see how they all become, they basically flatten out to form more of a planar structure. So the carbon-carbon bonds are really, really important in determining the shape of the other associated atoms and ultimately the shape of the molecule that these carbons are found within. Here are the most frequent partners that you will see bonding with carbon in a covalent fashion. Hydrogen, oxygen, and nitrogen are really found in the greater majority of these organic molecules. Other ones that will bind with carbon will include phosphorus and sulfur. Organic molecules that are made of carbon and hydrogen only are called hydrocarbons. But many organic macromolecules will have hydrocarbon components. They'll be made of other things, but really the bulk of what they are will be a hydrocarbon skeleton. Let me give you a great example. The fatty acids that we find in our fats and oils are modified hydrocarbons. Why are hydrocarbons really important to living organisms? Well, when you break those carbon-hydrogen bonds, there's a lot of energy that's released. So hydrocarbons are a great storage molecule for potential energy. A lot of the distinctive properties of our organic molecules are due to the functional groups that are attached to that hydrocarbon skeleton. So here's an example. We have a steroid hormone called estradiol, and we have a steroid hormone called testosterone. They are built basically of the same carbon skeleton. It's known as cholesterol. The difference, though, between cholesterol, estradiol, and testosterone are the functional groups. And as you change the functional groups out and change their number and change their arrangement, you will give each organic molecule a set of unique properties. This means that the functional group of that OH makes estradiol have a certain set of functional properties. And the functional groups, the ketone and the methyl group, gives testosterone an entirely different set of unique properties. There are numerous functional groups that are important in the chemistry of biological molecules, the organic molecules we're looking at. So here are the ones I want you to know for this course. The first one is called the hydroxyl group, which is OH. Now, in each of these groups, I have R, which is going to be the organic molecule, and the OH would be the hydroxyl functional group. So anywhere you see R, that's going to be the organic molecule, and then attached to it's going to be the functional group. The hydroxyl OH is a negatively charged functional group when you find it in aqueous solutions. This is because the H comes off as a proton, leaving a slightly negatively charged oxygen behind. And because of this, it introduces polar characteristics to the part of the molecule that it's attached onto. We have a carbonyl group, which is a C double bond O. We have a carboxyl group, which is a C double bond O OH. Again, it's a negatively charged functional group. Because of this OH group here, the hydrogen atom is going to come off, leaving a slightly charged oxygen behind. Again, like the hydroxyl group, it induces polar characteristics to the molecule. The amine group is an NH2. And in aqueous environments, this NH2 will pick up an additional hydrogen atom to become NH3+. So when it's attached to organic molecules, it is frequently found as NH3+ with a positive charge, thereby again introducing polar characteristics to that region of the molecule. Two other functional groups will introduce negatively charges and polar characteristics. That's the sulfate group, SO4, and the phosphate group, PO4. And then finally, we have a methyl group, which is a CH3, which is a non-polar functional group. It really doesn't change the characteristic of the molecule that it's attached onto, but it can be very important in the case of DNA and gene expression. We're not going to cover this in the course, but it does have functional consequences for when you attach it to things like DNA. I thought I would include this slide, which contains a very simple table that summarizes the functional groups that we just went over in the previous slides. You'll notice I also put these big red boxes around certain sentences where you can see particular functional groups will impart the characteristic of being polar or hydrophilic. The four organic macromolecules that you are responsible to know as a physiology student are carbohydrates, lipids, proteins, and nucleic acids. You can see from this pie chart on the right-hand side of the slide that about 30% of the human body's mass is made of these four organic molecules. The other roughly 70% is comprised of water. A lot of the organic macromolecules that I just listed in the previous slide are polymers. So let's take a step back and define what a polymer is. A polymer is a molecule made up of repetitive units called monomers. And polymers have very distinct shapes. They could be long linear structures. You can see two linear structures right here, two linear polymers right here. They could be branched. We have two polymers here that have a nice branching structure. You could take two linear polymers and attach them to one another, or multiple linear polymers and attach them. That's called cross-linked. And then you have these very complex three-dimensional net-like polymers that are called networked. Okay, a polymer is basically two or more monomers that you string together. To be honest with you, polymers can be very, very large, made up of hundreds of these monomers all strung together. Think of beads on a string. Well, how do we make these polymers? We have to link the monomers together. We have to form bonds between them. And the one thing that our four organic macromolecules
have in common is that they are all linked together, the monomers in each are all linked together through a reaction called dehydration synthesis. In dehydration synthesis, you have to remove a water molecule to form the bond. One monomer would donate a hydrogen atom and the other one donates an OH, as you can see here on the right-hand side. So here we've got this one, which is donating its hydrogen. We have the unlinked one donating the hydroxyl, and you have your molecule of water. This forms a bond between the polymer and an unlinked monomer and makes the polymer grow by one. When I was in chemistry way back in, the 1800s, dehydration synthesis reactions were also called condensation reactions because when they happened in a test tube, water droplets would condense on the inside of the test tube. Now, does it make sense to you that if you form a bond through extracting a water molecule, that if you want to break that bond, you got to put the water molecule back? I hope so, because in order to take off a monomer from a polymer, you have to break that bond and you have to put the water molecule back. Water goes back to, again, create the unlinked monomer, the one you're taking off with the hydroxyl, and then the shortened polymer has the hydrogen atom sticking out. This type of a reaction, which breaks up using water, is called hydrolysis. Hydro for water, lysis for breaking.
Even though carbohydrates are only about 1% of our body weight, they are a really important source of energy to our cells. We can store them as a reserve energy supply in the form of what is called glycogen. Outside of animal cells, if you take a look in plants and in fungus, even in bacteria, carbohydrates are a source of building materials that are used to build certain cell structures, like the cell wall of a plant, or the cell wall of a fungus, or even the cell wall of a bacteria. Carbohydrates are built of carbon, hydrogen, and oxygen. They are hydrophilic, so they are water soluble. And the functional unit of a carbohydrate is a monomer known as a saccharide or a sugar. Now, saccharides can be classified based on size. They are the simple sugars, which are built of one saccharide unit, known as a monosaccharide, or two saccharides strung together called a disaccharide. If you have 3 or more saccharides strung together, that is known as a polysaccharide. I think in some nutrition textbooks they might call it a complex sugar or a complex carbohydrate. The simple carbohydrates, or your simple sugars, are known as your monosaccharides and disaccharides. Now, a monosaccharide is a single saccharide subunit in which the number of carbons are pretty low. So a monosaccharide can be built usually anywhere from about 3 carbons to 7 carbons. For example, in humans, the common monosaccharides are 5 carbon sugars called pentose sugars, and the 6 carbon sugars known as hexose sugars. Just as an example here, the monosaccharides that you would find in humans are going to be glucose, galactose, fructose, ribose, and deoxyribose. These monosaccharides are by no means unique to humans. You will find them in a wide variety of other organisms. In aqueous solutions, monosaccharides are not linear structures. They curve around to form rings. And here you can see the 2 basic ways to represent the ring structure of a monosaccharide. The first one is known as the molecular ring form. You will notice that the carbons are labeled. The rule is find the oxygen and then go to the right. So in other words, kind of go clockwise. The first carbon is found to the right of the oxygen, and then you go around the circle with the 2nd, 3rd, 4th, and 5th carbon. The 6th carbon of this structure kind of waves out there like a flag. Now you can do the molecular ring form. You can also take away the carbon numbering and even take away the carbons and do what is called an abbreviated ring structure. Chemists and biochemists and biologists will know that where you see those 2 lines come together and the hydroxyl and the hydrogen are projecting out, we know that there is a carbon here. So that is the abbreviated structure. Numerous organisms will have within their cells these monosaccharides, glucose, galactose, and fructose. Now, each one of these, they're monosaccharides, and each one of them has the same molecular composition, C6H12O6. So right there, C6H12O6. Each one of them has the same molecular formula. Compounds with the same chemical composition, with the same molecular formula, but different structures, are known as isomers. Now, galactose is an isomer of glucose at the fourth carbon. In other words, the only difference between galactose and glucose is the fourth carbon. You, of course, can see that fructose is very different, right? But glucose and galactose are pretty much identical. Now, if you want to know whether or not you have glucose or galactose, you're going to look at the fourth carbon and ask yourself this question. Is the OH group facing up or is the OH group facing down? If the OH group is facing up, it is galactose. If the OH group is facing down, it is glucose. And the way I remember this is a phrase, look up to see the galaxy. Galaxy, get it? Up, there you go. Nice trick, huh? Glucose and galactose can be found in alpha and beta forms. To figure out whether or not you're dealing with an alpha or a beta monosaccharide, look at the first carbon. The difference between alpha and beta glucose is the position of the hydroxyl group at the first carbon. The difference between alpha and beta galactose is the positioning of the hydroxyl group at the first carbon. So if we look at that first carbon and the OH group is above the plane or is facing up, we have beta form. And if it's facing down, we have the alpha form. So in this case, we are looking at glucose, the difference between alpha and beta glucose. The same rule applies for galactose. So it is beta if the OH group is facing up, and it's alpha if the OH group is facing down. And my trick for this is you bow down to the alpha dog. So when you're looking at glucose and galactose, you have to ask yourself two questions. First of all, what am I looking at, glucose or galactose? For that, you look at the fourth carbon. Then you have to ask yourself, okay, now that I know what sugar it is it the alpha or the beta form? And for that, you look at the first carbon. For fructose, there's no problem at all because it doesn't even look remotely like that. However, it still has the molecular formula of C6H12O6. To build a disaccharide, we take two monosaccharides and we link them together. The disaccharides that we're seeing in this slide are formed when we take two 6-carbon monosaccharides and join them together and to create a bond called a glycosidic linkage. To produce a glycosidic linkage, you're going to need dehydration synthesis. You take a hydroxyl from one monosaccharide and you take a hydrogen from another monosaccharide, remove it to form water, and then you have a glycosidic linkage. So this slide is showing the formation of a disaccharide by taking these two 6-carbon monosaccharides and linking them together. If these two monosaccharides are glucose, you have formed maltose. If you take glucose and link it together with galactose, you have lactose. And if you take glucose and link it together with fructose, you have sucrose. These are 3 very commonly found disaccharides in cells, including human cells. Now, I imagine there are other disaccharides that are linked by putting different monosaccharides together, but these are the ones that we are going to concentrate on. 3 or more monosaccharides linked together gets you a polysaccharide. Polysaccharides are formed by stringing together multiple monosaccharides. So it is a polymer of monosaccharides, where each subunit is that monosaccharide. Each monosaccharide is going to be linked through a glycosidic linkage, which is created through dehydration synthesis. And if you can build a polysaccharide by sticking a bunch of monosaccharides together, you can break it apart and then liberate a bunch of individual monosaccharides. If building the polysaccharide is done through dehydration synthesis, breaking it apart is done through something called hydrolysis. In a hydrolysis reaction, you put the water back that you took out, and that breaks the bond. Some polysaccharides serve as storage materials. In animal cells, glucose is stored as glycogen, and it's used as a future source of glucose and thereby a future source of energy. In plants, the alternate of the sort of analogous version of glycogen would be starch. So in plants, they string a bunch of glucoses together to produce a storage material called starch. In cells other than animal cells, you will see also that glucose can be strung together to form what is called structural building materials known as cellulose. Let's take a look at these storage polysaccharides known as starch and glycogen. They are stored by plants and animals as a future supply of glucose, and that glucose is basically the starting material for ATP production. So glycogen is the storage form we're going to find in animals. It is stored in the liver of animals and the muscle of animals. It is a very highly branched structure. Each one of those green things is the monosaccharide glucose. So multiple glucose monosaccharides are strung together through dehydration synthesis, and it forms a very complex coiled and branched structure. If you want to break it apart, you have to put the water back in. So if you put a water molecule back in, you will hydrolyze it and take off one monosaccharide. This is catalyzed by an enzyme called glycogen phosphorylase. Starch tends to be a little bit more simple in comparison. So the simplest starch you would find is just a coiled structure of glucose known as amylose. But you can take amylose and modify it to be slightly branched, and that's called amylopectin. Again, just like glycogen, you can peel off this thing one glucose at a time. Enzymes will put the water molecule back and kind of break off a glucose monosaccharide. The enzyme that would break down amylose is called amylase. Our salivary glands of human beings make a large amount of amylase. In humans, amylase is also made by the pancreas. Amylopectin, I believe, is broken down by the enzyme called amylopectinase.
Cellulose is a polysaccharide of beta-glucose monomers that are found in the cell walls of plants. The linkage between glucose monomers and cellulose differs from what you might find in starch or glycogen. Cellulose is a very linear polymer, whereas things like starch and glycogen will form these coiled helices and branch points. And the reason why the linkage is very different is because cellulose is made up of beta-glucose monomers that are linked together, and starch and glycogen are made of alpha-glucose monomers. So again, this beta-glucose monomer sticking them all together makes a nice linear polymer. To build cellulose, you take a bunch of these beta-glucose monomers and you link them together to form what is called a cellulose molecule. The cellulose molecule is then bundled together. Several of them are bundled together through hydrogen bonds to form what is called a microfibril. And it is this microfibril that we then weave together with other sugars to build the cell wall of the plant. In the figure down here, you can see we have 1, 2, actually 3 cellulose molecules. These are then going to be linked together by hydrogen bonds to form the structure called the microfibril, and it is this microfibril that is found making up the cell wall.
In humans, we can't digest cellulose. It's known as an insoluble fiber. But what it does is it scrapes the lining of our GI tract, specifically the large intestine, and causes the production of mucus by the large intestine, so that we get a smoother transport of waste through the colon. But again, it is digestible by bacteria because they make the enzyme called cellulase. So I guess if you wanted to digest cellulose, you could take these bacteria, you could introduce them to your large intestine and hope that they will take up residence, and then you could start chewing grass. So what this slide shows you is it just shows you the 3 types of carbohydrates. So it's a really nice slide that just basically breaks down what we've covered over and sort of puts it in a nice organized schematic format. Okay, here's something called a medical application. every now and then I'm going to take a physiological topic that we've studied and I'm going to try and relate it to real life, or more specifically, a real life medical situation. I do this because I want you to understand that physiology is directly applicable to medicine. I also want you to stop and go, huh, cool, I didn't know that. Here's the thing about these medical applications. You're going to see them from time to time. There's one I'm going to talk about now. I think there's a couple of them in the next mini lecture. I want them to be fun and enjoyable and informative. I don't want them to stress you out, so they are not something that I test on. I just want you to come away from these medical applications going, huh, I didn't know that. So here's a medical application that has direct ties to polysaccharides. It's called high fructose corn syrup. High fructose corn syrup is any corn syrup that has undergone enzymatic processing by bacteria to convert some of the glucose monomers in the polysaccharide into fructose. And the reason why this is done is that fructose is a sweeter monosaccharide in comparison to glucose. So high fructose corn syrup tends to be sweeter than regular corn syrup. High fructose corn syrup was first described in 1957 by Richard Marshall and Earl Couey. They figured out a way to actually convert some of the glucose monomers into fructose and make a sweeter corn syrup. Unfortunately, it required the use of arsenic to make in large quantities, and that's not something that, the Food and Drug Administration is going to go for. I think the Food and Drug Administration was around in 1957. Now, in 1961, the commercial production of high fructose corn syrup was perfected by a gentleman known as Yamanaka, Dr. Yamanaka. Now, we to this day manufacture numerous types of high fructose corn syrup. You can see them listed there, and you can see their contents. High fructose corn syrup 90 is actually 90% fructose. It's actually used to create things like high fructose corn syrup 55 and high fructose corn syrup 42. Why do we use it? Well, it's cheaper. It's cheaper to use than sucrose, and it's used to sweeten foods. And the reason why it's cheaper than using sucrose is that back in the 1970s, I believe, import tariffs on sugarcane were imposed by our government. The United States generally got away from growing sugarcane, I think, towards like the beginning of the 1900s, although Hawaii still, to this day, I think there are still some sugarcane farms that are in Hawaii. But towards the middle part of the 1900s, more towards, I think, the 1960s and 70s, U.S. government decided to make it expensive to import sucrose or import sugarcane so that sucrose could be produced. High fructose corn syrup became the alternative sweetener to be used in foods. As a matter of fact, it was first added to Coca-Cola in 1984. And the reason why it's used, not only is it cheap, but because if you can chemically modify it to add more fructose, you get a sweeter corn syrup. On the sweetness scale, fructose is given the number 173. Don't ask me where these arbitrary numbers came from. Sucrose is given the number 100. Glucose is given the number 74. But look at corn syrup. It is not sweet at all. I don't know if you've tasted corn syrup. It's really not that sweet. It's a good thickening agent, but it's not very sweet. So if you can take the glucose of corn syrup and turn it into more fructose monosaccharides, you can triple the sweetness. Now, I'll tell you one thing about the United States. We're darn good at growing corn. We have more corn than we know what to do with. So guess what we do with it? In addition to turning it into ethanol, we turn it into high fructose corn syrup, and we use that to sweeten our food. Now, nutritionally and medically, there really is no difference between high fructose corn syrup and sucrose. They're both sweet. They both probably have similar health effects. I mean, there's articles out there that are saying, look, sugar is sugar is sugar is sugar. Doesn't matter if something is sweetened with sucrose or high fructose corn syrup, they are both equally bad. I'm not so sure about that. because the typical American weighs 25 pounds more than they did 25 years ago, and high fructose corn syrup entered into the American diet around 1975. Take a look at this chart. This chart compares and contrasts sweetening agents, sugar, which is sucrose, honey, high fructose corn syrup, and corn syrup. Again, not going to test you on this. I'm just giving you this so you can go, huh, I didn't know that. A lot of the facts you'll see in this slide were based on a YouTube video that was produced by University of California Television. It's called Sugar, the Bitter Truth. I don't know if it's still on YouTube. You can take a look for it. It was fascinating when I watched it. Did you know that today the average teenage boy eats and drinks 275 calories more every day? The average adult takes in 187 more calories. The average teenage girl takes in 335 more calories now than they did, 20 years ago. And if you realize that 1 pound of weight is about 3,500 calories, it doesn't take too many days to accumulate enough calories to put on a pound of weight. Now, the extra weight of the average American does not come from the fact that we eat fat. As a matter of fact, the calories that we get now from fat are decreasing because everybody's all about low fat this, low fat that. Yet obesity in the United States is climbing significantly each and every year. And this is because, really, if you go back to that teenage boy, 228 of those extra 275 calories that he gets every day comes from a carbohydrate. And in those that carbohydrate may in fact be high fructose corn syrup. Recently, in the last, I think, 10 years, there's been a 41% increase in soft drink sales and a 35% increase in what are the fruit drinks or the, those aids like Gatorade and what do they call them? What's the other sport drinks that are out there? The ones that are like the obnoxious neon colors. I can only think of Gatorade. Do you realize that 1 can of Coca-Cola has 150 kilocalories to it? And if you drink one of those a year, you can gain 15.61 pounds. All because of the sugar content, which isn't sucrose anymore, it's high fructose corn syrup. I'm not sure if this is right, but from this YouTube video, I learned that the average American ingests 141 pounds of sugar every year, and 63 of that comes from high fructose corn syrup. Let's just take a look at Coca-Cola. In 1915, a Coke bottle was 6.5 ounces, the little teeny tiny ones that you can kind of go and buy at Whole Foods and you go, oh, isn't that cute? That was the original Coca-Cola bottle. In 1988, the 44-ounce Big Gulp was introduced. Drink one of those every day and you'll take in 57 pounds of sugar every year. Studies actually now link increases in BMI, body mass index, and type 2 diabetes directly to an increase in soft drink consumption. So I'm going to leave you off with something called the Coke conspiracy. The Coke recipe is probably one of the most highly guarded secrets in the world. I believe there are only 2 people who know the recipe for Coca-Cola. They themselves, I do not believe, know who they are. They are not allowed to travel together. You're talking, the identity of these people is more closely guarded than, state secrets. But one thing the Coke recipe we know does contain is caffeine. Now, what caffeine does is it makes you pee. It also contains salt, so it makes you thirsty. So you know why the sugar is there? The sugar is there to hide the salt. That is the only reason why Coca-Cola is sweetened the way it is. And that's the way any cola or any soft drink actually is sweetened, so that you can't taste the salt, but the salt is there to make you thirsty. And the more Coca-Cola you drink, the more high fructose corn syrup you will bring in. The more high fructose corn syrup you bring every year, the greater chances are that you will see an increase in your weight. Now, there are countries that still sweeten their soft drinks with cane sugar. Mexico still does, and I gotta tell you, there really isn't a whole lot of things that are better when you're thirsty and it's hot out than a nice Mexican Coca-Cola. But again, there really is probably no difference nutritionally and medically between a Coca-Cola sweetened with sucrose and a Coca-Cola sweetened with high fructose corn syrup. So just be aware of that the next time you go to this grocery store or, you know, the 7-Eleven or a gas station, and you're thirsty and you pick up a Coca-Cola. All right, guys, we'll see you back for the next mini lecture 2B. In that lecture, we're going to look at lipids, proteins, and nucleic acids.